Okay, here we go. We are in Unit 2 now. So this is kind of like a really basic intro to genetics. This is not something that should be brand new to you. Everything in this PowerPoint should kind of remind you of what you learned in ninth grade. So hopefully this is like an easy, quick and dirty little like review, and we're not going to have to spend much time on it. So starting off, just as a first look into genetics, okay, we know that DNA exists, we know what it does, but this idea of genetics and heredity, and this idea that like we are able to pass on, well, genes, we didn't know the word genes, but we were able to pass on traits, that was discovered years, like 100+ years before we even knew what DNA was. So genetics itself is this branch of biology, right? It's under the umbrella of biology, but it specifically studies heredity, which is just the passing of traits from parents to offspring. And you can observe this in any organism. Obviously, we focus on humans because humans are pretty self-centered and they like to know about themselves. But you can study this in plants, animals, humans, bacteria, anything. I'm sure you've heard this name before, Gregor Mendel. He's the guy. He's it. He's the #1 dude. He's an Austrian monk. He was born in the early 1800s, and his background is in agriculture. His parents were peasants, so he grew up around agriculture and became an expert botanist, which is someone who studies plants. So that is kind of like the reasoning why he started to choose pea plants. We'll get more into why he chose pea plants in a minute, but he was interested in it because he knew it. Why learn something new when you already know something, right? So he's called the father of genetics because he was kind of like the first person to come up with all these inheritance laws, but he also was the first person to really write it down and be famous enough. I'm quoting famous because he's not really famous, but he had the money because he was a monk to like show his work to other people. So he gets all the credit for it. Being an Austrian monk in the 1800s is very different to what it is, like what you think of now. Back in that time frame, if you were a monk, it meant that you were well educated and you could do these types of experiments because you had money to do these types of experiments. So, like I said, he uses pea plants. He was very meticulous in his records, almost too meticulous in his records. There's definitely some controversy over whether he like fudged his results to make it a little bit more aligned with what he thought, but they're true. His laws are true. It's just whether he fudged his results to be like a little bit more accurate than what they were to prove he was right. So either way, the laws that he created are accurate. They've been tested time and time again and they come up true. He used statistics, and really in this time frame, this was like very exciting. This was like the coolest thing because now there was like actual answers. They knew that this was happening, right? You could see that it was happening, but now they knew why it was happening. Sort of. They haven't figured out DNA yet, but it was on the right track. So I keep saying he experimented on peas, but like peas are so lame. Like, why don't you study like, I don't know, cows or like flowers or anything cooler? Well, peas are like really easy to study. The male and female parts are in the same flower, so you can just basically self-fertilize, which is wicked beneficial. We'll get more into that in a bit. So because they were male and female parts in the same flower, made it easy to control. Peas are wicked easy to grow, they grow super fast, and they produce tons of offspring, so you can repeat experiments very frequently. And the garden peas have two clearly different forms, right? It's like yellow or green, wrinkled or round, purple or white, right? All of these are like very distinct forms. So you don't have to guess on whether, this pea plant is showing this characteristic or the other. It's just really obvious. So these are the 7 traits that he studied, right? He studied two things about the seed, whether it was wrinkled or round, whether it was green or yellow. The flower could either be purple or white. The pod that the pea seeds came in could either be constricted, which is like wrinkled or full, green or yellow, and then the stem, right? They either were short or tall, or they had terminal or axial flowers. And what that means is terminal just means on the end, and then axial is kind of like in the middle. So not on the ends of the branches, but really like closer to the stem. Now, monohybrid crosses. This is like your basic Punnett square. Mono meaning one, hybrid meaning cross. So, very aptly named. So Mendel did a bunch of monohybrid crosses just to see the outcome. So crossing two parents and only looking at one trait. That's what monohybrid means. So in this case of the picture, you see that he is combining, or he's looking at the purple and the white flowers. So he's combining those and looking at what the offspring are showing. Today, or I guess whenever you're hearing this, but in class, we're going to start with monohybrid crosses, so one cross, just to make sure that you guys still remember how to do Punnett squares. They're not hard, but we're going to build on them, so I need to know that you actually remember what you're doing. And then we're going to get into dihybrid and trihybrid, which is obviously a little bit more intense. You're crossing two traits or three traits, so it gets a little bit more challenging. Now, I mentioned this earlier about self-fertilization, so this is kind of how Mendel was able to figure out what traits he was starting with so that he could figure out what the outcome was if he knew what the parent genotype was, right? He knew what alleles, what genes the parents were passing on. He was able to kind of see what was going into the offspring. So by using self-fertilization, you can true breed plants, right? You can just breed them with themselves, so you just basically make clones of themselves. So this kind of just gave him a little bit more control over the traits that he was testing. So I just have this picture here so that you can see like this area right here, this orange area, it's called an anther. You don't need to know this, but this is just like good information if you are curious. This anther is where the pollen is, and pollen is the equivalent of sperm. So pollen is here, and this kind of green part that leads into this little like horseshoe shape, that's the female area of a plant. So the pollen from this anther can kind of just like go right over onto this area. It's called a stigma, and it's really sticky. So the pollen goes into the stigma and then goes down into the ovary, and then you can basically make your clones. So continuing on with what we were talking about, self-fertilization was used to determine like, okay, what are the characteristics of the original parent? If all the offspring are identical, then the original parent is true bred or pure or homozygous. Okay, so you can combine the male sperm and the female egg and look at what the offspring look like. If they are all clones, they're all identical, then it's pure. If they're not all identical, then the original parent is not pure, which means it's a hybrid, heterozygous. Maybe you've heard these terms before. So he would do this until he got all of these true bred or pure plants. By getting these pure plants, he could then use those pure plants as the parent generation or the P generation. And then he would know and be in control of what traits he was testing. So for his actual experiments, this is just sort of like a very brief overview. You have the P generation, which are purebred. And then when they combine, they make an offspring. That offspring is the F1 generation. F stands for filial. You don't need to know that, but if you're wondering, I'm just giving you all the information. So the F1 generation is kind of like the first offspring that are formed. And then that F1 generation self-fertilizes, so you just kind of combine itself with itself, and that creates the F2 generation. The F2 generation is just the second generation, and then you can kind of see that you have parents that are purple and white. All of the F1 is purple, and then when the F1 self-fertilizes, it breaks apart again as purple and white in a 3-to-1 ratio. And we're going to get more into that. We're going to practice with our ratios and everything, but hopefully what you're seeing is not brand new. So again, in the results, you can see all of this is like in the orange, that's like his actual results. You'll notice that they are extremely good results. Almost too good. Almost too good. So his results, upon reviewing this, you have a pattern that's showing. You'll notice that the ratio for all of these, wow, look at those great ratios, are about 3-to-1. So what he saw is that in the F1 generation, all of the recessive traits disappeared, but then in the F2 generation, they came back, and they came back in a ratio of 3-to-1. So his end result here, like his like final his like conclusion here, is that organisms carry both forms. It's just that one is dominant over the other, so one will mask the other. This is the law of dominance. This is one of his laws. So that was the first law. And now we're going to get into the other two laws. So alleles are the two forms. So like on the example we were just talking about, there's two forms of the flower. You could either be purple or you could be white, right? The flower color. And that are the two forms of the alleles. Alleles are inherited from parents, so from eggs and sperms. And when a parent makes sperm or eggs, their genes separate out. So each egg only gets one copy of each allele, and each sperm only gets one copy of each allele. This is another one of Mendel's laws. This is called the law of segregation, or the principle of segregation. And we'll talk a little bit more about this in detail in a slide or two. But when parents make their gametes, their sperm and egg, each individual sperm and each individual egg gets one copy of the allele. They either contain a dominant allele or a recessive allele. Depends on what the genotype is of the organism. Okay? So dominant alleles are always written as a capital, and recessive are always written as a lowercase. And you can see my example at the bottom of the screen, so you can look at like pea plants a little bit more. I'm not going to go over that. And then some more vocab before we like really get into it. This is just like a reminder. You should definitely have seen these terms before. This is not brand new information. So a genotype, and I've kind of said that in the last couple of slides, but a genotype is the genetic makeup of an individual. It's usually like when you write it, it's like the letters that you would write to represent that organism. So like capital T, or lowercase t. But you can also express genotype in words. And the words that you would use are, there's three options. You can be homozygous, which means that your alleles are the same, right? Two capitals or two lowercase letters. So you're either homozygous dominant, if you're two capitals, homozygous dominant, or homozygous recessive, if you're two lowercase letters. And then you can also be heterozygous, which means that you have one of each, so one capital, one lowercase. So homozygous dominant, that's one, homozygous recessive, that's two, heterozygous, that's three. And then the phenotype is just the physical quality. So what does it look like? It's tall, it's short, it's got brown hair, it's got blue eyes, right? The phenotype is what it looks like. Okay, I said that we were going to talk about his other laws, and here are the other two laws. So we've got the law of segregation, which we've sort of talked about already. So this is the idea that when you are creating gametes, So your individual sperm and individual egg, the organism's genotype is going to split, right? You only receive one allele. So all of your eggs and all of your sperm only have one copy of each of the organism's genotype. If you're looking at humans, right, humans have 46 chromosomes. That's just a fact, in general, okay? So if a human has 46 chromosomes, that means each of their egg and each of their sperm has 23, half of that, because they only inherit one copy of the allele. So they have 23, but the whole organism has 46. So the law of segregation just states that gametes get one copy of the allele. Now, Mendel's law of independent assortment. This is the idea that when you're looking at multiple traits, which is how all organisms work, right? Every organism has more than one chromosome. So when you're looking at multiple alleles, sets of alleles, they segregate independently of one another. So the law of independent assortment means that they are divvied up independently of one another. So if I have whatever trait A is, let's say this is hair color, and I have a second trait that I'm looking at, and that's eye color. So this is just an example. So hair color and eye color, they are going to divvy themselves up independently of one another. So I am not guaranteed to have brown hair and brown eyes. I could have brown hair and blue eyes. I could have blonde hair and brown eyes, right? They just totally are separate from each other. So it doesn't matter. Like you're never gonna, you can't force a capital A and a capital B to be inherited together because there are all these other options. This is called classical genetics. So this idea, the law of dominance, the law of segregation, and the law of independent assortment are your three kind of base pillars of what's called classical genetics. This is like, if you don't have any exceptions to any rules, these will always happen. And that's true. That is actually what happens. We are, of course, going to look at all the exceptions, so it will be a little different, but we'll get through it together. Okay, for our last bit, we're going to talk about Punnett squares. So this is the person who invented Punnett squares, Reginald Punnett. He lived in the late 1800s through the 1900s, right? Like mid 1900s. And his big thing that he studied was cows. So he everyone knew that genetics happened, right? They knew exactly what was going on. They knew that the parents would pass on their traits to their offspring, but they didn't know how. They didn't understand exactly what was happening. And so my man Reginald Punnett was like, oh, I got this. And he did a ton of statistical analysis and showed the exact patterns and what we now know them as Punnett squares. So he was the first person to kind of introduce Punnett squares. So Punnett squares are used to predict outcomes. It's a prediction. It is not set in stone. So this is just a prediction of what could happen. And so what happens is you have a parent cross, and each parent either goes on top, right? This capital T, lowercase t goes on top and splits. And then the second parent, little t, goes on the side and splits. And then, so each allele gets its own column or row. And then the alleles are just kind of like dragged and dropped into each of the boxes. So like this little t goes here, and this little t goes here. And then this capital T goes right below it, and this capital T goes 2 boxes below it, I guess. So once you kind of drag and drop all these alleles everywhere, you can see the possible outcomes, the predicted outcomes of each kind of parental cross. So for you guys are going to have to go through all of these steps when we're doing it. So step 1, determine the parental cross. Step 2, set up the Punnett square. Step 3, fill in the Punnett square. And step 4, determine ratios among genotypes and phenotypes. So we're going to go over one right now. So an example problem. We have pea plants. Round peas are dominant to wrinkled peas. And so we want to cross a heterozygous plant and a homozygous dominant plant. Heterozygous means one of each allele. So capital T, or I think I'm using T's here. If I'm not, I'm sorry. But a capital letter and a lowercase letter. And then a homozygous dominant is 2 capitals. So my parental cross, exactly what I said. Capital T, lowercase t, and then 2 capital T's. For step 2, we're going to set up the Punnett square. So I'm going to take my first parent and put it on the top, and my second parent and put it on the side. So now you see my Punnett square has been set up. And then step 3 is filling in the Punnett square. So for my first box, I'm going to drag this T over and this T over, and I get capital T, capital T. And I'm just going to keep doing that. So drag the alleles that are on the side, you drag them across the rows, and the alleles that are on the top, you drag them down the columns. Okay? Now, genotypic and phenotypic ratios. You only look at the 4 offspring. So in this case, I have genotypic ratios is where I'm looking at the letters. So I have 50% homozygous dominant, right? I have 2 out of 4 boxes showing big T, and 2 are heterozygous. Nailed it. I'm so good at genetics, guys. And then my phenotypic ratios are what do these look like? Well, because they all have at least 1 capital T, they're all going to show the dominant trait. So in this case, they are 100% round peas. Simple as that. So I've got some other examples for you, but I'm not going to go through them with you because this is part of your notes, so you guys are going to have to go through this. But I will help you out a little bit. So for this one, freckles are dominant, and so I want you to cross a woman who is heterozygous and a man who is homozygous recessive. So if we use the letter F, a woman who is heterozygous would be capital F, lowercase f, and a man who is homozygous recessive would be lowercase f, lowercase f. Again, I'm not going to go through all of this with you. I am going to practice Punnett squares with you in class, but if you are filling out the note sheet, you will need to do this on your own. Same with this deer who's dealing with albinism, which is a lack of pigment, right? They look white with red eyes. Homozygous dominant means capital letter, and albino is recessive. The only way to show a recessive trait is to be homozygous recessive. So little letter, lowercase letter, lowercase letter. And then finally, sunflowers, fuzzy leaves are dominant, so we have a true bred for fuzzy leaves. That means that it's true and pure, so capital, capital, with a sunflower that is heterozygous, so capital, lowercase. Our last bit is a test cross. This is just so that you are aware of what a test cross is. If an individual is phenotypically recessive, we know right off the bat what their genotype is, because the only way to show a recessive trait phenotypically is to be genotypically homozygous recessive. So the only way that you show being albino, for example, is if you only have recessive traits. The second you introduce a dominant trait, changes, right? Because dominant traits will always mask recessive traits. However, if we see a dominant individual and we know the phenotype is showing the dominant trait, we don't know if that organism is homozygous or heterozygous. And that's where the test cross comes in. So the test cross is done to determine if a phenotype of the dominant showing organism is homozygous or heterozygous. And so you will always cross in a test cross with homozygous recessive. You will always test in a test cross with a homozygous recessive. So in this case, you would test a homozygous, if you were testing an organism that you wanted to find out what their traits were, you would test it with a homozygous recessive organism, and then depending on the outcome, you would be able to tell what the other parent, that homozygous or that dominant showing organism, what their genotype is. So if they were homozygous dominant, all of their offspring would be dominant because the homozygous dominant genes would mask the recessive genes. If the organism that was showing the phenotype of the dominant trait was heterozygous, though, and you test crossed it, half of them, or about half of them, would show up as the recessive trait. So then you would be able to tell if the individual was heterozygous. Okay? That's everything for today. This is like a review, nothing crazy. You probably know all this, but we'll get into a little bit more serious stuff next time.